were to ask you, are you fond of gaining knowledge? Then this will do. A pleasant day, everyone. As of today, we prepared 10 published research, 5 in local and 5 in international, that will surely enlighten and help you to discover things. We all know that research is the process of collecting data, saving critical information, then analyzing and interpreting the data that is used for different purposes. And in a certain way, research is important in a field of works. A thought of hoping that you'll slump into your chair, take time to listen, and grasp a group of knowledge.
Everyone, on behalf of my group, I was tasked with reporting on the 5 international studies that were published and can be found on the internet. To begin with, the first study is entitled Lipids in Marine Ecosystem by Christopher C. Parrish that was published on February 13, 2013. This study was conducted in Memorial University of Newfoundland, St. John's, Canada. Sampling and optimization was the instrument being used in this study. The DOI number is shown in the screen. Let's now proceed to the strong and weak parts in the said study. For the strong part, it states that the researchers will study lipids having extractability in nonpolar organic solvents, which provides a convenient means of separating them from other compounds in an aqueous matrix. The extracts may contain multiple subclasses of both biogenic and anthropogenic origin. The researchers will try to determine production of biogenic material of dietary value to marine organisms as well as to indicate water quality, while the emphasis will be on biochemical and ecological aspects of marine lipids. The more stable compounds have also been extensively used in paleoceanographic studies. For the weak part, lipids provide the densest form of energy in marine ecosystems. They are also a solvent and absorption carrier for organic contaminants and thus can be drivers of pollutant bioaccumulation. Among the lipids, certain essential fatty acids and sterols are considered to be important determinants of ecosystem health and stability. Fatty acids and sterols are also susceptible to oxidative damage, leading to cytotoxicity and a decrease in membrane fluidity. The second study is entitled Fatty Acid Composition Analysis of Commercially Important Fish Species from the Pearl River Estuary, China by Zhang X, Ning X, Shi X, San X, Yu X, and Cheng Y, and et al. It was published on January 30, 2020, and this study was conducted at the Department of Agriculture and Rural Affairs of Guangdong Province and the Natural Science Foundation of China. And the methods, the instrument that has been used is experimental, and its DOI number was shown on screen. For its strong part, the researcher provides a large volume of available educational data which increase accuracy of the results that species from the Pearl River Estuary were generally characterized by high contents of saturated acids and low contents of polyunsaturated acids, which might be related to the dietary increase of diatoms and the decline of dinoflagellate during the past several decades. And the study provides a data that states the relationships between trophic levels and saturated fatty acids, monounsaturated fatty acids, omega-6 polyunsaturated fatty acids, and omega-3 polyunsaturated fatty acids of the marine fish species from the Pearl River Estuary in China, which become more understandable. The researcher also provides first description of the fatty acid profiles and lipid nutritional qualities of the economically important fish species in Pearl River Estuary, China, that helps in understanding the study. For its weak part, the only thing that can be noticed in the paper is that they use a first person, where in fact they should use third person, because first person can appear to weaken the credibility of the writer in research, as it reads as the writer's personal opinion. And the study needs a more further studies in order to make a comprehensive evaluation of the nutritional qualities of the fish species from the Pearl River estuary in China. And now, let's proceed to the third article, which is the effects of dietary lipid source and level on growth performance, blood parameters, and flesh quality of sub-adult olive flounder by Dong-kyo Kim, Kyung-dok Kim, Julie Yong-sho, and Sang-min Lee, that was published on June 2012 and conducted at the Aquafeed Research Center, National Fisheries Research and Development Institute, Pohang, Korea. And the instruments that was used in this study is sampling and experimental, and its DOI number is on screen. The strong part of this research is that the researchers who conducted the study were successful in their investigation of the effects of dietary lipid source and level of sub-adult olive flounder, growth performance, blood parameters, and flesh quality. They constituted a significant amount of educational data available, which improves the accuracy of the results. The findings of this study indicate that fish oil in fish meal-based diets for sub-adult olive flounder could be replaced by soybean and linseed oils without affecting growth or feed utilization, which will be helpful in formulating new fish meal. And the weak part is that the salt flow in this study is that no images of the sub-adult olive flounder were included in the research report. Where in fact, images of all kinds can be valuable in research report. This is because to the additional insight they can provide by supplementing verbal talks and also analysis. And now, let's jump to the fourth research article entitled Protein, Lipid, and Energy Requirements of Cultured Marine Fish in Cold, Temperate, and Warm Water by Jang Wang Kin, and that was published on 2013 and conducted at the Flinders University School of Biological Sciences, Adelaide, South Australia, Australia 2, and it uses sampling and optimization as the instruments of the research, with its DOI number on screen. The strong part of this research article is that temperature is one of the most important physical factors influencing fish growth. Under optimal temperatures, food energy partitioned into fish growth can be maximized. However, when a marine carnivorous species is cultured in an environment where temperature falls outside the optimal range of a fish, growth will be affected. The nutrient-environment interaction is important for optimizing a fish's nutritional requirements throughout the grow-out period. The most current global issue for the aquaculture industry is the inclusion of alternative ingredients into formulated diets to produce a sustainable seafood product. And the salt flow is that it affects the dietary protein requirement, including fish size, water temperature, feed allowance, amount of non-protein energy in the diet, protein quality, natural food available, and management practices. Moving on to the last research article that will complete the 5 international published research is the one entitled Protein and Nucleic Acid Metabolism as Proxies for Growth and Fitness of Oituna davisi, Earliest Developmental Stages by Lydia Yebra, Elisa Berdalet, Rodrigo Almeida, Veronica Perez, Albert Calvet, and Enrique Saiz, that was published on July 11, 2011 and conducted at Barcelona, Spain, which used sampling and optimization as its instrument and with its DOI number flash on screen. So, the strong part of this study is that the researchers' study contributed to the advancement of the study of small copepod nopalae physiology, growth rates, specific R's activities on RNA or DNA ratios, and their food saturating conditions were dependent on temperature. Somatic growth rates were calculated for either nopalae or copepodites. And the weak part states that the researchers finds difficulties on the incubation since it implies manipulation of the organisms and are time-consuming. As an alternative, biochemical or non-radioactive methods, most of them based on the estimation of protein or nucleic acid-related compounds, were proposed as proxies of the somatic growth. However, it was not until the last 15 years that such methods have experienced a major development with the availability of new highly sensitive and low toxicity proofs and assays. In general, the biochemical approaches constitute an instantaneous picture of the immediate past physiology of the organism and are sensitive, precise, and allow repeatability. And these were the 5 published studies found on the internet. An idea of hoping that you receive a deep of information from your time spent listening. But this video does not end here because the next reporter will be reporting on the 5 local research. So, Mr. Rolando Tingkal, please take your place.
Thank you, Daima. Reporting recently are the international published research which was accumulated through seeking in the internet. As for now, my part will be unleashed, voice-overing the 5 local research which will be started with the variation in chlorophyll A and B in the seagrass halojule in Central Visayas as the first information of the title which was published last October 8, 2013. They conducted this research in Bantayan Beach and Panela Beach in Dumaguete City, Negros Oriental in Tubuc, Siquijor Island in Central Visayas, Philippines, using test experiment as an instrument, while the DOI number will be attached on the screen later. Based on the analysis on the strong part of the research, the researchers generally attempted to observe the relative amount of chlorophyll A and B content of halojule unilaris and halojule pinifolia in Central Visayas, Philippines in relation to these environmental factors which are exposure, density, temperature, and light intensity. Analysis was run using a statistical software to identify trends or variances in the data. One way, ANOVA was used to determine significant differences of chlorophyll content among the sites, since light has been recognized as the main factor controlling the distribution and abundance of marine, estuarine, and freshwater flowering plants. Moreover, the analysis of the weak parts on this research is the failure of accomplishing the entire study. All material input and use might put to waste, and what's worse is, the researcher cannot provide proper results as well as conclude on what to do. Seagrasses are often the main primary producers in coastal ecosystem and contribute to maintain water quality by increasing the stability of sediments, biogeochemical cycling, and trophic dynamics. Additionally, spatial temporal distribution abundance and lipid content of colonid copper species in an aquilene area and an estuarine plum in northern Mindanao, Philippines was the second published research which was authorized by May L.J.P. Canencia and Ephraim B. Mitelio, which was published last October 8, 2013. Using experimental as an instrument, the DOI number will be attached in the screen later. They conducted this research in a aquilene area of Sindangan Bay and in estuarine plum in Butuan Bay and Himuon Bay. Based on the strong analysis, the researcher gave emphasis the reason why colonid cipapads were not found in some suckling stations due to migratory behavior and marine habitat characteristics and tolerance of environmental conditions, and they also provide reason why smaller individuals of all species appeared in the 2 shallow strata, while larger sizes were seen in the upper strata of dipstein and BGS, respectively, which gives the paper more accurate. Another strong part about this study is they provide a 3D image of the species and its characteristics and data that evaluate the difference between 5 species in different sampling sites. Nevertheless, the weak part of this study is that the fault in their data is so small that it becomes unreadable, and they explain the species individual by word and makes the study consume more pages. Some pages are laid out in landscape and needed to rotate your head for better understanding. The third published research, entitled Fatty Acids in 6 Molt Pelagic Fish Species under Crustacean Prey from the Mindanao Sea, Southern Philippines, which was authorized by Ephraim B. Metelio and Ana Arlene Asperas Eya, which was published last August 2014 using sampling as an instrument. They conducted this research in Mindanao Sea, Southern Philippines. The DOI will be attached later. Based on the strong parts of this research, the researchers' strength is that they were able to provide supporting and credible data on the study's finding. They provide information on the study of fatty acids in 6 small pelagic fish species under crustacean prey from the Mindanao Sea in the southern Philippines. The fatty acids depicted on the 6 pelagic fish species samples under crustacean food were supported by data. while the weak point is the identification of the 6 tiny pelagic species that comprise the sample. Researchers did not include the common name of each species, which would make it much easier for readers to recognize if it had been provided. Consistently, survival and settlement rates of Heliotis acinera larvae at different salinity levels were the fourth SEEK research, which was authorized by Gian Rose H. Makerang, Romeo D. Cotarao, G.C. H. Makerang, and Fiona L. Pedroso. This research was published last October 8, 2013 in Iloilo, Philippines as the area where they conducted to fully complete this research using test experiment as an instrument. Based on the strong parts of this research, the researchers determined the survival and settlement rates of donkey's ear abalone, also known as H. acinena, and different salinity levels. It also determined the optimum range of salinity for better larval survival and settlement of donkey's ear abalone. Haliotis acinena is one of the most commercially important tropical marine gastropods in the Indo-Pacific region. Furthermore, the weakest part of this is the very low survival and settlement rates of H. acinena from the veliger to plantigrade larvae during the first run was due to the addition of diatoms as feed, which was not anticipated to be centrifuged to remove excess water with high salinity. Having failure in the entire process just to accomplish the research is one of its weakest parts. Moreover, no mass mortality happened in each treatment. Upon studying the lipids in marine ecosystem, it may be thorny for the entire researchers to find a conductor to fully complete the entire process. Nevertheless, the result is incomplete and useful enough to provide a perfect outcome. The last SEEK research to fully complete the 5 were entitled Biology, Conservation, and Management of Nematopelimon Whips Fishery, also called as Aramang locally, which were authorized by Romeo C. Culasing, Lenemfa P. Mulena, Eunice A. Leogan, Simeon Arabanal Jr., and Loren G. Amog, published last October 8, 2013. They conducted this study in Apari, Cagayan River estuary in the Philippines using interview as an instrument. Based on the analysis about the strong parts of this research, the researcher found out that Nematopelimon whips, locally known as aramang, is an exotic palatable soft-shelled shrimp and endemic species in Apari, Cagayan. It is caught throughout the year. The fishery shows varied annual fluctuations for the last 5 years. The resources does not appear to be underfed or overfishing at present due to increasing production of catch. Based on the findings of the researcher, documentation of aramang fishing has established the most of the catch is dried and marketed locally and internationally. Moreover, the weakest part is that the researcher found out that there's an increased demand for aramang primarily due to its gustatory taste. In the late 19th century, the value of conservation among the Aparianos was inculcated due to a drastic decrease in supply as a result of unabated and indiscriminate gathering by fishermen, mainly for their daily consumption and as a source of livelihood. It resulted as being the weakest part of this researching, since the growth parameters of Nematopelimon whips should be studied for 1 year and further trials must be conducted to ascertain the benefit. The published international as well as the local research which were reported by us will end here. It's quietly a long report, but in behalf of our group, we are hoping that you grasp a group of knowledge which will surely abiding you as you grow. Moving on into the last part of this video, let us all witness and take time to listen the monologue of our groups.
My future research plan will be tackling about boosting the growth of fishes to cope up and provide solutions to the food security in regards with fish production. It would be probably accumulate group of fishes to complete the entire process. Moreover, I would be using articles which was published in the internet to support my research reliability. It would be surely useful not just in the country but also in the food security to boost the growth of fishes, since my conclusion as well as the recommendation upon accomplishing my success will provide proper acknowledgement supported with dependable evidence to support solution to the problem. How about you, May? What's your future plan?
Well, my future plan is that we need to preserve fish and other aquatic resources in order to prevent fish spoilage and lengthen shelf life. Fish spoilage and lengthen shelf life are designed to inhibit the activity of spoilage bacteria and the metabolic changes that result in the loss of fish quality. The future of aquaculture has significant potential in helping provide a healthy and sustainable protein source for future populations.
populations. However, to reach this potential, a substantial increase in production is needed to ensure future demands for protein are met. Oh, I also heard that you have a future plan, Rolando. What's yours?
As expected from a flabbergasting groupmates, interminably I will help. Conditionally, if given a chance in seeking and building my own research, I would probably study the life habitats of rare species on the ocean, as well as analyzing how their existence contributed into biochemistry. It actually are broad topics, so it will presumably takes time to complete and publish conclusions in regards to the entire process. It actually is an awesome study, since you will get to know some of the rare species, as well as seen them vis-a-vis, so probably you'll gain proper knowledge on how do they live. Apart from that, presenting an experimental analysis would be expected, since, as what I have said, it is too broad that it may take time to accumulate the possible results. Drowning down into the next person who will share her future plan, might as well share it once.
Definitely. I have a future research plan that I would want to share with you. My future study would focus on innovation and new farming technology in aquaculture for new species, to assist in increasing food production in order to secure food security. The aquaculture sector not only produces a healthy food product, but it also employs thousands of people globally. The aquaculture business clearly has immense potential to help provide a healthy and sustainable protein for future generations. But change is required to assure its sustainability. That is why I am much more interested in researching and discovering increasingly sophisticated farming technologies that will shape the pace of transformations needed for aquaculture to meet the demand. I hope you learned something from what I shared. So, let us move on to another person who has a future research idea to present.
Wow! You actually have a very good future plan, guys, and I myself learn a lot of things from you, but I won't allow myself to be left behind. So, to be the last woman standing sharing my own perspective about my future plan, it should be started with a comparison of the lipid composition and 2 or more marine fishes. I'm still in the process of thinking what species could it be, but it should be seen in the market. I am here because I want to elaborate why we need to study the lipid composition of a marine fishes, because fish oil have been a natural constituent of human diet and have been reported to be hypocholesterolic, which they contain essential fatty acids, especially oleic and linoleic acid, as well as other polyunsaturated fatty acids. As polyunsaturated fatty acids was being reported that it is being protective against cardiovascular diseases. Therefore, it means the diet rich in fish oil will be protective against cardiovascular diseases and possibly other diseases like cancers and arthritis. I want an answer for this study for the sake of the human health. As lipids perform 3 primary biological functions within the body, they serve as structural components of cell membrane, function as energy storehouses, and function as important signaling molecules. Conducting this study would help if those species could provide a good source of oil. And to end this, in behalf of our group, we would like to express our grateful thanks to our biochem teacher, Ms. Iris Magnayi, for the learnings, to our family for supporting us, and of course, to our Almighty Father who make this possible. And why would I forget to sacrifice and provide teamwork, right? Let us show them our beautiful places. Team, we filled as one fish. Thank you, everyone. To expedite the last, I'd like to leave in a quote. Although no one can go back and make a brand new start, anyone can start from now and make a brand new ending. Car brand.